So, talking about what is called as your microbial diversity. Now, the more we know about it, the more we discover, the more we explore. Probably, we are more what you call as it is more astonishing, and it is more surprising to know more about these organisms which are more in number. So, talking about organisms and their classification, we generally like to classify the organism based on their nutrition. And we say that whether an organism is autotroph or whether an organism is a heterotroph, right? Very rarely we come across organisms which can really change their mode of nutrition completely. Now, during their life cycle, actually, if they are shifting from autotrophy to heterotrophy or heterotrophy to autotrophy, this is somewhat like astonishing and surprising to know that there are such organisms which do exist. We have been talking about symbiosis. Lately, we have been talking about recently about your endosymbiosis, where an organism is living inside the other organism. Probably we believe that the origin of the chloroplast, the origin of the mitochondria is all because of these particular endosymbionts. But do we really know about organisms which are showing a temporary endosymbiosis, right? One such organism which was recently discovered back in 2000 and reported officially in 2006 in Japan is your Hatena. The name itself is like something different, something which is different, that in Japanese they refer to as what is called as your Hatena. So, talking about this organism that is your Hatena. Now, Hatena is an organism which is a unicellular protist. Now, it is actually a single round celled protist which has a heterotrophic mode of nutrition. It has two flagella, and with these two flagella it goes on hunting and ingesting and eating the food. Like that is what this Hatena does. So there is nothing special about it actually up till now. But things don't stop here. If you see and follow its life cycle, which these two Japanese scientists did, so they found that this particular organism, that is your Hatena, it is autotrophic at one stage. So it shows a life cycle like that of a plant, and it is heterotrophic at the other stage, like that of an animal. So this Hatena, as I told, has two flagella, and with those flagella, it is feeding itself. And while feeding, it comes across an alga, a green alga called as Nephroselmis. And this particular Nephroselmis, once it enters into this Hatena, there is a change both in the algae as well as the host. Now, this particular alga, Nephroselmis, acts as an endosymbiont. Now, once this alga has entered into the Hatena, Hatena shifts its mode of nutrition. Now, up till now it was heterotroph. Now it becomes an autotroph. Now, that is due to the virtue of this particular alga, that is Nephroselmis, because this Nephroselmis actually provides nutrition. Since it is photosynthetic, it now performs photosynthesis and lives as an endosymbiont with this particular Hatena. The more interesting fact is that this Hatena, it cannot reproduce itself unless it has this particular endosymbiont, that is your Nephroselmis. So once this Nephroselmis has come in, right, in the meantime this Nephroselmis also undergoes certain changes. Now it will lose its the flagella, it will lose its cytoskeleton, it will lose its Golgi complex, it will lose its mitochondria. What it will retain is some of its cytoplasm, it will retain its nucleus, it will retain its plastid, and that is what is required by this particular Hatena. It is found that the free living form of Nephroselmis and the endosymbiont form of Nephroselmis are also different. Now, this endosymbiont form has a plastid which is 10 times larger than the plastid which is present in the free living form. Now, obviously, this size is compensated by the reduction in the cytoplasm of this particular Nephroselmis. So, Nephroselmis has undergone these changes in itself. So, now it works only as, say, an organelle. But now again, it is not a complete organelle, because as we know that this organelle divides when the cell divides, this does not happen with this Nephroselmis. Some changes take place in Hatena also. As I told, the major change which takes place after it has engulfed this Nephroselmis or made this Nephroselmis as its endosymbiont is that it will change its mode of nutrition. Now from heterotroph, it becomes autotroph. It loses its flagella. that is the feeding apparatus actually. With the help of those two tubes, it was actually feeding itself. So it loses its entire feeding apparatus. So its feeding apparatus is lost, Hatena's feeding apparatus is lost, it becomes autotrophic, and now this engulfed nephrocellus, or now let me call this as a temporary endosymbiont nephrocellus, acts as an eye spot. And it helps this Hatena to move and locate itself towards light so that more photosynthesis could be performed. So it actually acts as an eye spot, and there is a positive phototaxis which is brought about with the help of this particular nephrocellus. Now, when this particular cell divides, as might be expected with other organelles, it does not happen. As I told, that this endosymbiont does not multiply inside. Now, when the cell divides, there are formation of two daughter cells. One of the cells is green in color because it has received this nephrocellus, whereas the other one, it remains white. So, this latter one, which has remained white, has not received the nephrocellus component, and therefore it is a heterotroph, and it will remain so until it gets its own species of nephrocellus. Now this again will have a flagella, it will have its feeding apparatus, and it will search for a nephrocellus, and it will not reproduce unless it gets acquainted or unless it engulfs this particular nephrocellus. So this intriguing interaction is really worth noticing and studying, that how exactly organisms are evolving. Like how exactly endosymbiosis is working even today. It was not something which was happening in the past, it is still working today. So we really need to explore much more of this microbial diversity. So thank you so much.